My wife Allison was diagnosed with Alzheimer's disease, must be now close to 15 years ago. I was quite surprised that we heard the same thing time and time again. Try 3 Advil a day, it might help her. And oh yes, there are some FDA-approved drugs. They treat the symptoms briefly, but they really won't help.
My mother had Alzheimer's, and by the time she was my age that I am now, she didn't recognize me or know me. My father cared for her, and it killed him before she died.
Alzheimer's is the biggest disease of our time. It affects old people. We're such a generation of wanting to be young, and yet the disease is such a burden to our society. It's a huge cost. $200 billion of Medicare and Medicaid cost for families with the disease, and they spend another $200 billion in private spending and lost wages.
Alzheimer's represents clear and present danger, not only to this country, but to the world. It's probably the biggest predictable disaster. If we know a disaster like that is coming, we can predict the size of it. We need to do something about it.
10 years ago, we met Rudy Tanzi, and he told us 3 basic things. That Alzheimer's was decimating the country, that there was no possibility at the time of a cure, and that the new genomics were yielding potential cures, but there was no money to fund the genomics. That caused us to make a basic decision. We had to find a cure for that dread disease.
And so we called Henry McCants, who I'd worked with before, and later brought in Phyllis Rappaport to start with our core group.
After meeting Rudy, I couldn't sleep that night. I felt that we needed a national intervention. I remember as a child getting the polio vaccine, and it was a nationally funded intervention, and I felt if we could have a prevention for Alzheimer's.
When I first got together with the founders of Cure Alzheimer's Fund, and we got together and said, we can do this. We can put up the funds and find the rest of these genes, because right now, for the first time in 2004, 2005, the technology, everything's there to do it.
I think the fact that we had the audacity to put Cure Alzheimer's in our name suggested we were just not about basic academic research. We have a very functional goal that we're dedicated to, which we allocate all of the funds that we raise to that end.
I've spent my career as a venture capitalist, and I've seen the power of really visionary entrepreneurs coupled with proper venture capital financing. My goal was more to really use a venture capital approach to change the speed at which scientific research, medical research was going to make progress on this disease. Cure Alzheimer's Fund actively goes out and seeks a dream team of researchers. We have scanned the country, we've identified who we think are the leading in Alzheimer's research and who are complementary to one another. And we've pulled them together under Rudy Tanzi's leadership to hopefully function as really a collaborating dream team.
The format that they have, where they have teams researching different areas around the country, this approach casts the widest net and you never know where a breakthrough is going to come from. Where if you do something differently and you support just one organization, you're losing a lot of potential brain power that you could have at your disposal.
We are purely philanthropic. We want all the energy, all the focus on finding a cure. Our founders and board members said that they would pay all of the expenses for the organization. That means that every dollar, every penny given by any other donor goes directly to research.
I think the approach of the Cure Alzheimer's Fund and the portfolio of solutions is in many ways an open source approach to be able to share this content with scientists and academicians all over the world so that we can break those barriers and be as innovative and as being as responsive as we can to expedite a solution.
What Cure Alzheimer's Fund can provide that government cannot provide is fast, focused, flexible funding on ideas that are great ideas that may not be funded by bigger entities.
Our approach to Alzheimer's is to start at the root cause to get to the genetics, which is why we began this foundation in the beginning.
Using our roadmap, we'll be able to predict who's at risk early on, and most importantly, empower them with the ability to prevent this disease from striking in the first place, based on their genetic risk.
I'm very proud of the work we are accomplishing. People are joining us in big numbers. We're getting thousands of people funding us through the internet.
Cure Alzheimer's Fund is, I believe, in its format, the most effective format for fighting a terrible disease.
We're at an inflection point on cracking this disease. It's a very exciting time.
10 years ago, it was unimaginable to us that we would have the success we are having with the Cure Alzheimer's Fund. Please join us in our quest. Hello, everybody. My name is Jeff Morby. I'm chairman of the Cure Alzheimer's Fund. I am here with the rest of our associates to celebrate this 10-year anniversary of the founding of the Cure Alzheimer's Fund. And I'm here to wish you happy Alzheimer's Fund Day, as articulated by the mayor of Boston. This is a day of celebration for all of us because three families 10 years ago got together, and we were all dissatisfied with what was happening with Alzheimer's. We'd all been touched by Alzheimer's, and there had been no cure in 100 years. The government was mediocre at best in funding research, and the new genomics were yielding potential cures, and the funding of new genomics was not occurring. And so we had the audacity, as Jerry said, to say we can solve this problem. And we had a secret weapon. Secret weapon was Rudy Tanzi. And we made a deal with Rudy. We said, Rudy, if you put together the finest team in the world of Alzheimer's scientists, we will fund you in your research. And so we made a deal, and we got started. And 10 years later, you see the results of this initial collaboration. So I would like to just introduce our founding members and just say a couple of words about each one of them, and then proceed to the next step. So first of all, I'd like to introduce you to Jackie Morby, my wife, and Henry McCanns, our associate. And then Phyllis comes later. The reason I mentioned Jackie and Henry is because they were both venture capitalists, and they speak the same language, and they were quite instrumental in helping us articulate the venture philanthropy concept that we've implemented so successfully. Phyllis Rappaport is an amazing person. She's very imaginative, conceptual. When we first started the Cure Alzheimer's Fund, we had another name for it, so complicated that I can't remember it. And Phyllis said, you know, we should be saying what we want to do. We want to cure Alzheimer's. So let's name the foundation the Cure Alzheimer's Fund. And so that's what we did. And Phyllis has been all along very conceptual and innovative and creative in her support of what we're doing. Now, more recently, we've been honored by the entry into our board of two very outstanding people. One of them is Bob Greenhill. Bob, will you stand up, please? And the other is Matthew Zulik. Matthew, where are you? These are two very seasoned businessmen who I think will make great contributions to what we're doing. Bob has already been on the board for 3 years, and he's a man with great wisdom. He's a legend in the industry, and he's a man of terrific insight. And by the way, his son also comes to our board meetings, and he's pretty smart too. Apart from the person just mentioned, I would like to then thank some other people. First of all, I want to thank Rudy, but I'll talk about him later, but I want to thank all the members of our research consortium and our scientific advisory board. Most of them are not here, but one of them, David Holtzman, has been a major supporter of our activities for since inception. David, where are you? Right here. Could you stand up, please?
You forgot Matthew.
Then I also would like to make special mention of Tim Armour. Tim Armour is our president and CEO. And without Tim and his team, we would be nowhere. Tim, where are you? Stand up. Finally, I'd like to thank all of you and our other supporters for your significant support over the years. We have more than 20,000 families and other institutions that are supporting our activities. And this has really provided us with the raw material with which we can fund our activities. And so that's the extent of my thanks. Now I would like to introduce a very important and wonderful person, Charlie Collier. Charlie has contracted Alzheimer's disease. He was the head of philanthropy and wealth management at Harvard. And we have a short video about Charlie, and then his successor, Alistair Halliday, will make a few remarks about him.
I was diagnosed with Alzheimer's. My challenge is that every year, even with the drugs that we have available, my functioning is getting worse. But I'm a fighter, and I'm leaning into this challenge.
Part of Charlie's Alzheimer is his aphasia, word blockage. He has trouble retrieving words.
I've come to the conclusion that all of us will have challenges in our lives. We are all dealt a hand of cards in life. Some of it is good, some of it is not so good. The best way to think about it is this: how am I going to react to the cards that I've been dealt?
Charlie spent his professional life helping families grapple with difficult questions. That's why he wants to do this. He wants to talk about it, to help other people think about the questions, think about their lives in looking at this.
When my friends and family hear about my volunteering, they typically don't know anything about Alzheimer's or about the fund, but I'm talking it up.
Charlie has leaned into this. He will talk to anybody. He will engage in a conversation.
Every minute, there's one baby boomer that's been told that they have Alzheimer's. The country needs to wake up to this.
There are many ways that one can introduce Charlie Collier. I could tell you that he is a family man in every sense of the word. We know from Charlie's book and from his career that he's passionate about understanding what makes families tick, how families work. But I also know that Charlie has been a dedicated and loyal husband and father and son and brother and partner. I could also tell you that Charlie is a graduate of Phillips Andover and Dartmouth College and Harvard Divinity School. Or I could tell you about his pioneering work in the field of wealth advising, where Charlie did things that nobody had ever done before. I could tell you about his career as a groundbreaking fundraiser and about the Harvard Medal, one of Harvard's highest honors, which the university bestowed on Charlie a few years ago for his wonderful service to that institution. I could also introduce Charlie as a spokesperson for the Cure Alzheimer's Fund and a brave soldier on the front lines of that terrible battle. But I would prefer to introduce him by talking about the qualities that, in my opinion, have enabled Charlie to carve such a remarkable life and career. When I think about Charlie, three words come to mind. The first is conversations. Charlie has a deep and abiding belief in the power of family conversations to heal families in times of estrangement and transition and misfortune. And in fact, the power of conversations to help families at all times. The second word is courage. Many of us have an instinct to turn away from difficult conversations, difficult family issues. Charlie has the opposite instinct. Charlie's instinct is to turn toward those conversations, toward those difficult family issues, to march right at them and confront them with transparency and with courage. And the third word is caring. Charlie is able to do all of this because he cares about people and he cares about their families. Charlie has been passionate for decades about helping families think through how to live well in the face of challenging issues. When that work was taken from him, he redirected that passion to helping families think through how to live well in the face of Alzheimer's. Conversations, courage, and caring. Before I invite Charlie up to the podium to give him a gift from the Cure Alzheimer's Fund, I'd like to read a letter that was written to Charlie by our former U.S. Treasury Secretary and former president of Harvard University, Larry Summers. Larry wrote this a few years ago upon hearing of Charlie's intent to retire from Harvard. Charlie, I know I speak for everyone at Harvard in thanking you for your remarkable service to the university. Your wisdom, human sensitivity, and loyalty have made the Harvard community a much better place. The resources you have helped mobilize for the university are the least of your contributions. You have helped so many be fulfilled in some of life's most important dimensions. I will always be grateful for all the support you gave me while I was at the Harvard president, and for the wisdom you shared with my family. In ways that might surprise you, I often find myself quoting things that I learned from our conversations. No one should have to face the challenges that you're facing. And certainly, in a fully just world, it would not be you who was so burdened. But I know of no one with more fortitude and grace in facing adversity than you. Please know that you will be very much in all our thoughts going forward. And if there's anything that I can ever do for you or your family, please do not hesitate to call. Warmly, Larry. Conversations, courage, and caring. Ladies and gentlemen, Charlie Collier. Come over this side. I want what you do. How about that? And there's a big bow tie in the middle of it. A Paul Revere bowl.
There's one other friend that we want to thank, and that's Senator Markey. Senator Markey was going to be here today, but he obviously is engaged in all kinds of activities and he couldn't make it. But we decided instead to have a brief interview. I went to Washington to sit down with him, and we're just going to show that right now.
Hello, Senator. Jeff, thank you for having me with you today.
Well, it's a pleasure, and I'm here to just brief you on what we're doing with Alzheimer's. First of all, I want to thank you for your outstanding support of our activities over the last 10 years. Your leadership in the Congressional Task Force on Alzheimer's and your sponsorship of the National Alzheimer's Project Act and your multiple activities that have supported the Cure Alzheimer's Fund. We honor you for that.
Thank you so much. My mother passed away from Alzheimer's. Everyone who has an Alzheimer's patient in their family knows how profound that is. And I've been working since she passed away in 1998 towards raising awareness here in the United States Congress. I created the Alzheimer's Task Force back then. But ultimately our goal has to be to find the cure for Alzheimer's. And so thank you for all of the work you've done in creating an environment where the cure for Alzheimer's is now something that is increasingly on the minds of every American.
Yes, it's also getting closer and closer. Our focus has been on one thing, to find a cure, because that's the core problem. And so over the last 10 years, we've been very successful in bringing this research to the point where we're really on the verge of moving to a new stage.
Well, I think that our goal should be, and you're the leaders in this, is that we reach a day where children have to turn to the history books to find that there ever was such a disease as Alzheimer's. And we can do it.
Yep.
If we can put a man on the moon, and that's what President Kennedy challenged America to accomplish when I was a boy, well, the next mission is to the mind.
Right.
Because that's the last uncharted territory on Earth, the minds of human beings. And this effort to find the cure is something that I think is going to increasingly be driven by the kinds of research that you are helping to foster.
That's true, and I would say our moonshot is the next stage we're about to enter into. We just completed the first ever whole genome sequencing of Alzheimer's disease ever done. We now have a wonderful database of basically all of the genes of Alzheimer's. And we're now moving into kind of the final stage, which is what we call genes to therapy. So we've identified 20 of the most important Alzheimer's genes. And gene by gene, we are now developing therapies to stop them from doing what they're doing. And so that's our next phase, and that should result in substantial therapies.
Well, that is fantastic. Research is medicine's field of dreams from which we harvest the findings that give hope to families that there will be a breakthrough. And what you're doing is putting together all of these clues through the mapping of the genetic code that will give us an opportunity to have the smartest people in America, in the world, then find that breakthrough that gives hopes to families all across the planet.
Yeah. Well, what we have done is we've developed what are called intervention points. Alzheimer's progresses in stages, and so there are various stages where you can intervene. So each of these points is where we're focusing on developing a therapy. And I think that will really make a difference for researchers around the world, because they will now know these intervention points. And our activities are now supported by over 20,000 families. Wow. Give you an idea of the number of people that are supporting what we're doing. So we're very excited. We're very excited.
Well, in the same way that we created a team to eradicate smallpox, to find the cure for polio, that's what you're doing right now.
Yep.
And you need it to be as broad and deep as is possible, and I just can't congratulate you and thank you enough for all of the work which you do.
Well, thank you very much for your support.
Thank you, sir.
Now I'd like to present you to Rudy Tanzi. All of you know of him now. He is one of the real pioneers in Alzheimer's research. He was one of the first to discover the first 3 Alzheimer's genes, and he's been leading our activities. So I won't talk any more about him because you want to hear him. Rudy?
I really like that kind of introduction, Jeff. That's, because otherwise you have to sit through and hear what you did your whole life, and you feel like you're already, gone. So I want to thank our founders, our board members, our Cure Alzheimer's Fund staff, all of you here, our supporters, all of you watching on the internet, for celebrating our 10th anniversary. It blows my mind to think it's been 10 years and think back at how much we've done and how much of an impact we've already made on Alzheimer's disease. I think the key to Cure Alzheimer's Fund is bang for your buck. We're lean and mean, and we take the money that comes in and we fund only the highest impact projects leading to big papers and really trying to affect how we think about this disease. And I'll talk later a little bit about the finding that we just published in the journal Nature this week that was covered in the New York Times and throughout media around the world, which is very nice timing. Somehow Tim Armour has all these connections to get the New York Times and Nature and Newsweek and Time to do all this for our 10th anniversary, so we'll never underestimate Tim. But this is just wonderful to have everyone here. And what I'm going to do is talk to you about where are we in terms of understanding Alzheimer's and where are we going next. And I honestly think that we're in the beginning of a new era of Alzheimer's research, and a very optimistic one. And I'm hoping that by the time I'm done with this lecture, you will know as much as anyone about this disease and how it's caused and what we need to do to stop it. And one of the themes I'm going to use, and I actually got this term from my colleague at Harvard, Risa Sperling. I love this term of right patient, right time, right drug. And this is something we've been talking about at Cure Alzheimer's Fund for over 2 years now, that we really have to think about Alzheimer's and its stages as a 30-year disease where pathology is accumulating 15 to 20 years before symptoms. I think Dave Holtzman, my colleague on the research consortium, an MVP on our consortium, has shown that better than anyone, that this is a disease that takes about 30 years And you have to use the right drug at the right time, including preventing the disease way before symptoms even occur. So that's going to be my goal, is to teach you about that this afternoon. But let me tell you a little bit more about the Cure Alzheimer's Fund. The objective from the very beginning, as stated by the founders, was to fund highest impact research and aimed at successfully treating and preventing Alzheimer's. As you heard, our founding families pay all the expenses. Every dollar in is a dollar out to highest impact research. And it was my pleasure to put together this dream team of researchers who are really the most proven. If you look at their curriculum vitae, they all have hundreds of papers, dozens of seminal paradigm-changing papers under their belt, and have all come together to really break down the silos, work together, collaborate, email together, talk together. One thing the Cure Alzheimer's Fund did, in addition to everything else they did that was great, was to get these people together and get us talking more than ever. And that's why I love the comments you made about conversation. It is about, Charlie, it is about conversation. It's about caring, conversation, and courage. I think that's a great motto for our research consortium, because that's what it's all about, having the courage to take chances, do big things, conversations about how to do it, and like everyone here, caring. I mean, everyone is here because life is nothing unless you're serving. It's all about service. That's what we're doing in research, that's what you're doing supporting what we're trying to achieve. So I really appreciated those comments. Just some facts about Cure Alzheimer's Fund. We've raised $41 million to date, 15,000 donors. I think I just heard 20,000 families on the video, so more than that now. 27 million distributed, with reserves now ready to go for genes to therapy over the next year. 84 projects, 115 grants, 150 papers, I think it's even more than that now, cite support of the foundation, with 7,600 citations, high impact papers. 58 laboratories at 30 institutions in 14 states, and also including labs in Germany and Israel, and we're trying to extend more into international funding as well. And this is because Alzheimer's is such a terrible disease. 5.4 million victims in this country, 3.5 million deaths in the last 10 years, 13 million family members impacted, 15 million unpaid caregivers, plus the emotional burden, and worldwide, 40 million people have this disease. That will go up to 100 million or more by 2040. It's an epidemic that is just going to get worse, because all of modern medicine basically has us living longer. And the fact is, lifespan right now is close to 80 years old. And at 85, 50% of the population are either have this disease or are starting this disease. So you just do the math and you can figure out things are going wrong. And this is a chart I've shown many times. It's showing the mortality rates for various diseases from 2000 to 2010. And you can see nicely AIDS, pneumonia, stroke, heart disease, diabetes, cancer all down, Alzheimer's way up in red. And then look at the NIH research dollars in millions. Alzheimer's is the red-haired stepchild. For some reason, Alzheimer's gets much, much less than the other major diseases that threaten healthy aging. So we need a wake-up call. We really need a wake-up call in the government and in the budget to say Alzheimer's is going to take us out. This is a graph that Jeff Morby put together, calculating, if you think about how much, if you look at how much of Medicare and Medicaid Alzheimer's disease already consumes, then you consider 74 million baby boomers heading toward risk age. At what point will Medicare and Medicaid be basically catastrophically impacted by Alzheimer's? It's somewhere between next year and 2020. So if we don't do something about this disease, it can single-handedly collapse our healthcare system, collapse Medicare and Medicaid. So it's very important, in addition to everything we do here, to keep telling our congressmen and our president, we need more support to stop this disease, or it's going to stop us. Now let's get to the disease itself. Many people ask, what's the difference between dementia and Alzheimer's? Well, Alzheimer's is a type of dementia, and it's the most common form of dementia you see in the elderly. 5.4 million patients in this country. And the brain pathology of Alzheimer's begins in almost everyone after 40 years old. So this is a disease, like I said earlier, that's taken root 20 to 30 years before you even see symptoms. And now, thanks to biomarkers and imaging strategies, looking in the brain, the pathology through imaging, looking into spinal fluid for biomarkers, a lot of the work that my colleague Dave Holtzman here has been involved with and is pioneered, we can tell if someone's on their way to this disease way before symptoms. We can start to do that. But it's important to then empower that person with therapies so that the disease is stopped in its tracks and doesn't take root. So that's our goal, early diagnosis and early intervention. And with genetics, we can also even do early prediction, so one knows if they have to be more vigilant throughout their life about getting early detection of the disease. I mentioned this fact already about over 85. Our current drugs are basically better than nothing. They treat the symptoms, drugs like Aricept and Namenda and others, but they don't stop the disease. They don't prevent the disease from progressing. They're not nearly good enough given what we need to do. Risk factors for this disease, number one is age, family history, gender, women get it more than men, head injury, stroke, even emotional trauma has been implicated as a risk factor. I think the common denominator is as the brain begins to take insults with age and nerve cells start to die, it could be a head trauma, it could be a stroke, it could even be cortisol levels going up after loss of a loved one or a traumatic event in one's life, that as the brain undergoes insult and injury, this can start to trigger the disease, and as you start to develop pathology, as I'll show you, it can spread throughout the brain progressively. So I mentioned the current therapies that are basically better than nothing. They basically try to enhance cognition, but they don't stop the pathology. They don't stop the neurodegeneration in the brain. Well, what can we do? These are just a few things you can do. I think number one is physical exercise. At least take a brisk walk every day. I included basketball here, which I play on a weekly basis, although now I'm limping with a torn medial meniscus. And based on the number of bangs to the head I take now under the boards, I'm thinking that's probably not good for me. So I'm staying on the perimeter now, trying to get my three-point shot a little better. Healthy diet, Mediterranean diet's been shown to be useful, fruits, veggies, olive oil, less red meat. Social engagement. Not staying home, but actually being socially engaged, getting back to conversation. A couple of herbal remedies that are being worked on, ashwagandha and cat's claw. I think these two have probably the most interesting data for effects on pathology, and we're looking into these more closely now. Learning new things. Coming to a lecture like this is actually protecting you. You're learning new things. It's better than brain games, you know. Brain games teach you how to do one strategy, but it's really when you're learning new things and making new synapses and reinforcing those synapses you already have, you're building up your synaptic reserve. And in the end, Alzheimer's disease is about losing synapses. So I tell people, well, when you're planning for your retirement, you want to save money and you want to save synapses. You need both, okay? So anything you can learn is good. And doing crossword puzzles and Sudoku is not going to be good enough. Also try to reduce your emotional stress. We're working on the effects of meditation and yoga and its benefits for the brain. A lot of work going on in that area these days. Get enough sleep, and I'm a huge hypocrite, by the way. I do not practice what I preach, but I can preach it. 8 hours of sleep. You need to get that deep, what's called slow wave sleep or delta wave sleep. It's been shown in some studies that this is when you consolidate your long-term memories, so you're learning all this stuff all day, and then the short-term memories get consolidated into long-term memories during deep sleep. And also it's during deep sleep that you clear debris from your brain. A lot of this gunk that accumulates in your brain, I call it mental floss, getting deep sleep. And a nice study out of University of Rochester showing that the brain gets rid of a lot of that material, that gums up around the nerve cells during deep sleep. And you need to get enough sleep to ensure that slow wave deep sleep. So what about the pathology of the disease? And these are the original drawings by Dr. Alzheimer when he first described what he called a miliary body. We call it a senile plaque or an amyloid plaque. And he said the inside of the nerve cells were choked with these tangles that seem to kill the nerve cell from inside. These are still the two major hallmarks of the disease. And much of the research that's going on is to understand how do you form these plaques? How do you form these tangles? Are the plaques linked to the tangles? And I'm going to address much of that later on. This is just another artist's rendition of what a plaque might look like. And here's a nerve cell that's been in contact with this amyloid, and now it's forming the tangles inside. It should look like this down in the corner, the healthy neuron, but in here it's full of these tangles. So what we also know is that the pathology of Alzheimer's disease spreads. So although it's starting in one part of the brain, as you form these tangles, they actually spread. And much of this work was actually first shown at WashU St. Louis. Dave Holzman's colleague Mark Diamond first showed that this is a spreading pathology. So if you think about some of these folks who have head injuries, an NFL football player who had too many concussions, or a soldier in the battlefield who suffered from a bomb blast, this is enough to form some of these tangles, and then as they form, they can start spreading throughout the brain. So I think of it as the tangles being like a fire that spreads throughout the brain. Then the question has to be, what causes that fire? And most people who are going to get tangles later, what's causing those tangles? Is it the amyloid buildup? Is it bangs to the head? Is it a stroke? So we have to think about this disease. I like to think of it as a lighting the match and starting to fire. The tangles are the fire, but what lights the match? What lights that fire? So before we get into that more deeply, one of the questions I get often is, what's the difference between Alzheimer's disease and just getting old? Just having a senior moment. And I'd like to use this cartoon here. Caution, senior moment in progress. This is the one mostly for me. I know I came into this room for a reason. And then the two dinosaurs sitting on this little island as Noah's Ark floats away saying, oh crap, was that today? So that's what really happened to the dinosaurs. And this is a real set of signs. Someone said to me, Senior Center, don't forget. Senior Center, remember to turn. Senior Center, wake up. Senior Center, lunch only $5. Senior Center, turn now. And what this brings home is that as we get older, frankly, we don't pay as much attention. As we get older, we go from, when you're a child and the whole world is magical, every moment's a wow moment. And as you get older, it comes into every moment being a so what moment. Been there, done that, yeah, uh-huh. And this is the issue, is that a lot of our issues we have with memory are actually due to attention, not paying attention to what you're doing, not really registering where you dropped your keys, not registering what you were going to do or who you were going to call on the phone. How many of you ever called somebody on the phone and had that moment of panic where you have no idea who you called? Raise your hand. Because I do that. It's just not paying attention. But then when do these moments become pathology? That's the issue. So what you need to know is that the way the brain evolved is that memory is reinforced by emotion. Memory is reinforced by emotion, by passion. Little kids learn like crazy so fast because they're so passionate about everything going on. And as we get older, we might be too busy, too much bandwidth, we might get jaded, we might become apathetic, but that's what you have to get back. And I think a perfect example of memory reinforced by emotion is, if you're old enough, how many of you remember exactly what you were doing when JFK got shot? Because I remember. How many of you remember when the space shuttle exploded? I remember I was buying ski poles. How many remember exactly what was going on 9/11? So these incredible traumatic events just hardwire those memories instantly. You can remember every little minute of what was going on. It's just a great example of how emotion and passion dictate memory. And we need to keep doing this as we get older, even at subtle levels. Be passionate about your life, pay attention to what's going on. But you know what, sometimes that's not enough. That's not enough to stave off Alzheimer's disease. And Alzheimer's is a very different situation. You are developing pathology in the disease. You're losing over time your sense of self. You've built up this whole life, your personality, your behavior, your predispositions. People know and love you for who you are. And little by little, that whole tapestry of who you are gets ripped out by this disease. So there can't be anything worse than that. And this is why I think all of us are here, that we find it unacceptable. It's an unacceptable disease. We absolutely have to stop it any which way we can. So in Alzheimer's disease, what's happening first is short-term memory is really being affected. And you have basically, I'll just put the slide up, but I'm going to explain it instead. That you have all these things happening, right? So you're taking in sensory information. You're seeing things, hearing things, smelling things. That information then comes into a part of the brain that then sends it all at once to your short-term memory in an area called the hippocampus. Hippocampus means seahorse in Greek. It looks like this, like a seahorse. And one of the first things that happens, and this is my colleague Brad Hyman showed this back in the mid-'80s, is that that cable that sends that new information to your hippocampus to store it for short-term memory, then later that will become long-term memory if it's repeated, is that cable gets broken. That what's called the perforant pathway that sends that information starts to break down. So you may be present and taking in sensory information, but it's not getting stored in short-term memory. So what you start to lose is continuity from minute to minute, from second to second, right? And it's this continuity that gives us a sense of self, that we have memory and we're even remembering what happened a few minutes ago till now. If you lose that continuity of time because you're not storing information, because your brain's not letting you, this is when you start to lose this sense of self. This is the problem, and this is what we have to stop. And this is why using therapies that bring back self-awareness, like music, arts, things that you're really passionate about throughout your life, you can use these to help bring back your sense of self-awareness. So just to illustrate this is Alzheimer and his patient, Lois Alzheimer's and his patient, August Dieter. And in 1906, she was 55 years old and was admitted to an asylum in Bavaria with psychiatric issues. And she was sitting on her bed with a helpless expression. This is from Dr. Alzheimer's own notes. And he said to her, "What is your name?" She said, "August." He said, "Last name?" She again said, "August." "What is your husband's name?" She again said, "August, I think." "How long have you been here?" 3 weeks. She had actually been there for 2 days. And then at this point, he says she broke down and cried and kept saying, "I've lost myself, I've lost myself." And this is why this disease is so terrible. It is a loss of self, not only for the patient, but for the loved ones and those who have grown up to love this person and is now losing them day by day. So again, to me, there's nothing worse than this for the patient or for their loved ones, and this is why we need to stop this disease. So what do we learn about ourselves from Alzheimer's? Well, sense of self requires memory. You need to have context in space and time of what's happening around you. When that starts to go, you start to lose that sense of self over time. So very late stage patients eventually lose this sense of time and space and thus self. What are we going to do about it? And this is where we get into the Cure Alzheimer's Fund roadmap. And our roadmap begins with the foundational studies at the genetic level, find the genes involved, because those are like the givens in a long algebraic equation, do the translational research to understand how those genes cause the disease, and then drug discovery and drug development to capitalize on that information and being able to empower people with the ability to stop this disease. So if we just look at the history back, from 1906 to 1986, we knew the pathology of the disease. There were plaques, there were tangles, there's also inflammation in the brain. But then starting in 1987, we and others found the first Alzheimer's gene, the amyloid precursor protein, which I was able to name back in 1987, the presenilins, which we co-discovered with Peter Hislop and Jerry Schellenberg, and then ApoE, the late onset gene. It was found at Duke University. These first 4 genes taught us the very first things about this disease. And genes are so important because they really teach you what's going on. So they allow you to think about the possibility of predicting the disease early, detecting it early, and preventing it before it strikes. This is ultimately what we want to do to eradicate Alzheimer's, is never let it strike in the first place. If you take what you learn from these genes, this gives you drug targets. This allows you to think about therapeutic intervention. So I think a great example of this is heart disease, right? Back decades ago, Brown and Goldstein, who got the Nobel Prize for this work, they had a family with very rare genetics where they got heart disease in their 40s or 50s. And they found that the mutation that they had was in a gene that was involved with controlling cholesterol. And they proposed that cholesterol might have something to do with heart disease. And then going forward, now we know that to prevent heart disease, we have to have the right diet, exercise, weight control, if necessary, statins like Lipitor or Crestor or others to keep our cholesterol down. So in Alzheimer's disease, we have an analogous situation. In Alzheimer's disease, it's A-beta, or amyloid beta protein, this little protein that's made in the brain. And likewise, like heart disease, we can think about controlling A-beta with diet, exercise, weight control, or if necessary, drugs, but we don't have the drugs yet. So A-beta accumulates in the brain and it starts to form little clumps that get in the way of synapses and communication between nerve cells, and they form these plaques that also accumulate outside of nerve cells that we say are toxic to the brain, but it's been controversial. Is the amyloid really the toxic material that starts the disease, or is it just a marker? Is it just a side consequence of aging? And I'm going to address this when I talk about the work that came out in Nature this week that you've heard about. So the amyloid can be present in the senile plaque, and here in this very core of the plaque that's sitting outside of nerve cells, that's amyloid. It can be present as these fibrils that look like spaghetti that make up that core if you look really closely, or these little blobs on this side that are called oligomers, and these are little blobs that can sneak into the synapse and cause short circuits in neurons, nerve cells that are trying to communicate with each other. So this is how we think amyloid works, and all four of those first genes we found had in common that one way or another, they increase the accumulation of beta amyloid in the brain. Now, what we also know is that amyloid's not enough. You have to have tangles, and then that has to lead to nerve cell death. Then these tangles spread through the brain. And then as nerve cells die, brain inflammation is activated, and actually probably kills many more cells than the original pathology. See, the brain, inflammation in the brain has good intentions. Your brain has a very primitive immune system. It's not quite above that of an oyster or a horseshoe crab, okay? The brain doesn't have antibodies and T cells and B cells and all the fancy stuff you hear about with our regular immune system. It's very simple. In the brain, we have these little cells called glial cells, and they're constantly checking things out. They're supporting the nerve cells. By the way, glia in Greek means glue. And the glial cells are nurturing the nerve cells, they're maintaining them. So they're usually like housemaids. But they have this other side to them. If they get upset, they can become SWAT team soldiers, okay? So what's happening is that as nerve cells die, they first take on the same role as the Red Cross or National Guard. They come in, they clean up, they feed people, they take care of the situation and nurture. If too many nerve cells die, they have a little conversation, well, I'm just picturing this, and they say, wait a minute, this many nerve cells dying, something's wrong. And they make the same assumption they made 25,000 years ago when we lived till 25 years old and ran around in the forest. This many nerve cells dead must be an infection, must be a bacterium or a virus or a fungus. And so now they no longer are helping, now they think they have to fight a foe, a pathogen in the brain. So they start shooting out oxygen-based bullets, free radicals. Friendly fire, collateral damage is killing more nerve cells. They start shooting out little molecules called cytokines to get the other types of glial cells excited, and now they start fighting. Astrocytes get excited. They can start chomping up the synapses. So you see what's going on is almost like it's an autoimmune disease of the brain based on this primitive immune system, but what triggers it is the original pathology and nerve cell death. And now from genetics, we're learning all the different factors that hit amyloid, the tangles, and I think most recently, how that threshold is set in each person in terms of whether your glial cells are going to stay Red Cross, National Guard and help, or throw their hands up and become a SWAT team and hurt. So this is a really important message I wanted to give you. So these are the things that go on in this disease, and genetics is teaching us how this all happens. So I like to think about, don't worry about the stuff in the middle. I like to think about this disease as the earliest stage being the amyloid beta protein that gets made. It starts to accumulate with the help of metals like copper and zinc. In fact, without copper and zinc, you can't get accumulation of A beta based on our experiments and many others. As the amyloid forms, you start to see tangles. Again, it's been controversial. Does amyloid really lead to tangles? But I have that in my diagram. As nerve cells start to die, the brain reacts with inflammation, and you get this vicious cycle of more pathology and more cell death, because the inflammation itself becomes like an insult to the brain. Remember, it's insults that get this thing going. So the question is, if the tangles are spreading and causing the cell death, what causes the tangles? In the amyloid hypothesis, it's proposed that amyloid does that. But you can also see tangles forming, if you have 5 very unfortunate seasons in the NFL with too many helmet-to-helmet hits and concussions, you can get tangles without the need for amyloid. One bomb blast in Iraq or Afghanistan, you can get tangles. The question is amyloid a very slow, decades-long insult that leads to tangles the same way, and then the tangles spread, start the fire, then inflammation does the final knockout punch on the brain? That's the question we're asking. So in the very first therapies that were developed based on those first 4 genes, remember those first 4 genes were leading to more amyloid, right? So I've often used this analogy of the overflowing kitchen sink, like the brain overflowing with amyloid. And what we've seen is some companies have tried to turn down the production of the amyloid, in other words, turn off the spigot. These were gamma secretase inhibitors. They failed because they weren't safe. Gamma secretase is an enzyme that helps make the amyloid beta protein get produced in the brain, but it has a lot of other jobs in the brain. And what we learned is you can't hit it with a sledgehammer. You need the enzyme, and there are many unsafe adverse events if you just block that enzyme. We've also seen trying to clear A beta from the brain. We've seen immunotherapies that have come and gone. This is where you have antibodies that are made to amyloid beta protein, and the antibodies trigger the clearance from the brain through a variety of pathways. But all of these trials failed. Well, we know why the gamma secretase inhibitors failed. They just weren't safe. The antibodies probably failed because we were doing this too late. Remember, I told you the amyloid is starting 15 to 20 years before. We're treating patients who already have the disease. So along the lines of this idea of right patient, right time, right drug, amyloid drugs were being used most likely too late. You have to use them a lot earlier. And I think this comes down to this figure that comes from a paper by Risa Sperling. I think this figure's been shown in many different presentations. And what you can see here is in the red line, that's amyloid. And see how the amyloid is coming up preclinically, before symptoms. By the time you get to that first dotted line of mild cognitive impairment, the amyloid has gone up and it's already starting to plateau. Kind of like heart disease. You've already had your high cholesterol, you've already made your coronary artery disease and plaques. Now you're going to be at higher risk for a heart attack. So treating amyloid in someone way over here on the right who's already suffering from dementia is kind of like having a patient who just had a heart attack go to the doctor, and the doctor says, here, just take a Lipitor. You had to do that 15, 20 years ago. Or here, just have a better diet. Well, yeah, you want to do that. If we can hit amyloid, you want to do that in a patient right now, but it's not enough. You have to think about these other events. The tangles are coming up next, the inflammation is here, and dementia. So these are the stages of the disease, and a big point of my presentation today is match the right drug to the right stage. So this doesn't mean that amyloid's not a good target. We just have to hit it earlier, we have to hit it better. So I've listed here some of the other ways now that we're thinking, we and others in academia and in pharma are thinking about how to hit amyloid. You can hit the other enzyme that makes amyloid called beta secretase. And beta secretase inhibitors are being developed by Merck, Eisai, a bunch of different pharma companies, another attempt. We hope for the best here, but again, beta secretase has other targets in the brain. I'm a little worried that you can't hit beta secretase with a sledgehammer either. You might have to be a little smarter. And where we were a little smarter recently is on gamma secretase, where instead of inhibiting the enzyme, we're modulating the enzyme. And this is work that Cure Alzheimer's Fund was brave enough to fund, work that I'm doing with Steve Wagner at UCSD. We've been developing drugs that stop that gamma secretase but only for its effects on APP, the amyloid precursor protein. In other words, we're letting gamma secretase do all the rest of its thing, but only stopping the gamma secretase from hitting the amyloid precursor protein. So it's very specific for A-beta. And those drugs, the very first generation of those drugs that Steve and I worked on 15 years ago, just got FDA approval for phase 1 clinical trials. So we'll see those go in. Then the newer versions of those drugs that we think will be even better, Cure Alzheimer's Fund supported, we're now applying over the next year for FDA approval for the next round. So we're going to have 2 different versions of the same drug, hopefully in clinical trials, one about to go in, another one about to go in for application. And again, this wouldn't have been possible without Cure Alzheimer's Fund. The other type of therapy for clearing A-beta, the antibody that's still in play is from Lilly called solanezumab, and actually David Holtzman all but invented that technology, so that's why he's on our dream team, our research consortium. And solanezumab, I have here not with a red X through it, because it actually had efficacy in the mild patients, the patients who were still in the mild stages. If you sort them out in the trial that was done, they had some improvement, enough so that there's a trial going on I'll tell you about for prevention. Other immunotherapies in play are Biogen IDEX, and we'll hear about that probably sometime this year. And then another drug that clears amyloid from the brain is from a company that actually I started in my lab back in 1997, a company called Prana Biotechnology with a drug called PBT2. I'll tell you a little bit about that one too. So this is where we are with amyloid. The A4 study is following up again on this idea that solanezumab had some positive effects on the mild stage patients. And Risa Sperling over at Harvard, Brigham and Women's, Mass General, is doing a prevention trial where she's taken folks who are normal, they don't have the disease, they're between 65 and 85, but you can see by imaging that they have lots of amyloid beta protein in their brains, so they're at higher risk for the disease. And then asking over a period of 168 weeks, can solanezumab make a difference versus placebo for affecting whether they go on with that much amyloid in their head to the symptoms of Alzheimer's disease. So we're waiting to hear about that. And there's about 1,000 in that trial with 500 placebo, 500 drugs. So we'll hear about that sometime later. Now, another way to do this, as I say, is the GSMs, and Steve Wagner and I have our GSMs that Cure Alzheimer's Fund supported, now ready to go into phase 1, 2 rounds of them ready to go into phase 1, starting the first trials just for safety. So we'll keep track of how those do. And then finally, the Prana drug, PBT2, in its first trial back in 2008, it successfully reduced amyloid beta protein and the phospho-tau. This is what makes up the tangles when you look in CSF. And they also had positive results on improving executive function, which is a type of cognitive domain in memory. So this was very encouraging. And then more recently, they did a small trial to ask whether the drug could affect amyloid burden in the brain. This was only 42 people. And what happened, if you look to the right, is in red, PBT2 actually dropped the amount of amyloid in the brain. It was a very nice effect. It was significant. But somehow, the patients who didn't get the drug, who got the placebo, also, what I think is a fluke, had amyloid drop as well. And I say it's a fluke because here in the bapineuzumab trial, amyloid goes up in the placebo group over time, and the amyloid stayed the same, more or less, in the treatment group. Here, the treatment group went down, but the placebo group also went down. So as a result, the trial was negative, but because it's kind of a fluky result, it doesn't make me lose my hope, and also looking at the future for this drug. So if we think about these together, we can think about hitting amyloid early, preclinically, before there's dementia, using them like statins in heart disease, solanezumab, gamma secretase modulators, beta secretase inhibitors, small molecules like PBT2, to try to hit the disease early, and maybe one or more of these could become a statin of Alzheimer's disease. So the next question is, if we know that amyloid is building up early, and we have candidates as statins, will pharma do these trials? Will pharma do a prevention trial that can take 5 years if you really want to do it right and cost well over $1 billion? Well, what pharma has to know, and the question they're going to ask is, is it really worth stopping amyloid, right? Does amyloid really trigger this disease? And although that's been the hypothesis for 30 years now, since George Glenna first described the amyloid beta protein, it remains debatable. So this is where I want to get into the story that broke this week in Nature and New York Times. Does amyloid actually lead to tangles and the rest of the disease? Because only then I think pharma will start to have confidence to say, yes, it's worth doing long, expensive trials to block amyloid early on and see if we can stop Alzheimer's from taking root. So if we take the same diagram, we can ask the question, do tangles really follow amyloid deposition? And I'm going to show you the work of Se-Hoon Choi, who's a postdoctoral fellow in my lab, and Do-Yeon Kim, who's another principal investigator in the unit I direct over at Mass General. And I wanted to show this slide from Einstein, who said, no problem can be solved from the same level of consciousness that created it. And I show this because up until now, most of the studies that were done to ask questions have been done in mouse models. And in mouse models, this is what we do. We take Alzheimer's genes, we put them in mice, and we ask, can this somehow block tangles? And if you have amyloid, do you get tangles? Well, it turns out in mice, you put in these Alzheimer's genes that make amyloid, and unlike what happens in the human, you don't get the rest of the pathology. You don't get the tangles. So what my colleagues did in our unit, and this was the study that was covered in the New York Times, this is Do Kim here, was to try to create an Alzheimer's in a dish model. Try to do this using human nerve cells in a dish and seeing if we could recreate the pathology and ask the question, does amyloid really start this disease off? Does it really trigger the tangles? Big question if you're going to think about hitting that early on to prevent this disease in the first place. This is the paper that just came out in Nature, and I'll show you what we did. So originally, many studies were already taking nerve cells of some type, nerve cells from mice, or in some cases, nerve cells that you make from stem cells of humans, and they would grow them in a petri dish in liquid, okay? And in these little red squiggly things, the amyloid beta protein, and they'd float around in the liquid, but they wouldn't really form the pathology. You wouldn't see the plaques, okay? And Do-Yeon Kim had a very simple idea, and it was based on a very simple fact. The brain's not made of liquid. The brain's gel. The brain is 3 pounds of Jell-O in your head. It's not made of liquid. Maybe in some sci-fi movies it's made of liquid, but not in reality. So what he did was create a 3D system where he used a gel that mimicked the brain with certain proteins in it that mimicked the brain environment, and said, let's do the experiment in this gel instead. And what he saw was that in addition to just the little amyloid beta proteins floating around by themselves, now they start to form clumps. They actually start to form plaques, the same way you see in a brain. Simple idea, thinking out of the box, and the results were that you could now get plaques to form if you just did these experiments with human nerve cells created from stem cells, but do it in the same medium that's the brain, which is gel. And what he could see was these wonderful neurons that were growing, and then this is just showing you the 3D aspect of it. This is showing a thin layer of gel, and you're seeing the nerve cells that are growing in this gel, and they're forming, see all these axons and dendrites? They're forming connections. The one on the left is the low mag, the one on the right is a higher magnification, and you're actually seeing these nerve cells growing in this gel and communicating, forming networks with each other the same way you see in the brain. So it's like, great, this is great. We're actually mimicking nerve cell networks in these little gel-like Alzheimer's in a dish assemblies. And then, well, how do I get out of this? Ooh. He didn't tell me this was going to be tough. There we go. Phew. And then what we saw was just an astonishing day for us as we looked in there, and I say we, it wasn't really me, it was the guys doing the work, Se-Hoon and Do, and they could see plaque. This little orange guy is a plaque forming in the gel in the nerve cells. And what you're seeing here is basically a serial imaging through the plaque. And I think this might be a movie. Yeah. So here you can, if I play this movie, and I know how to do this, you can see now going through the plaque from the top to the bottom. So it's a 3D plaque sitting in those nerve cells in the gel. So for the first time, we were able to actually form a plaque in a dish. And now what we could do is we could test whether our drugs, and this is one of the gamma secretase modulators that Cure Alzheimer's Fund supported, and say, in this system, can we add our drug, gamma secretase modulator, and get rid of the plaque? And sure enough, we could. This is where we don't add the drug, and you're seeing here these plaques among the nerve cells. Add the drug here, and you see no plaques. So what we do is we grow these nerve cells for about 6 weeks, let them form their networks. By 6 to 8 weeks, they're going to form the plaques, but when you have the drug, they don't form the plaques. So now we can test for drugs that stop plaque formation in 6 to 8 weeks in a dish, rather than what would take 1 year and a lot more time and money in mouse models. So that was great. That was step one. The next question was, how about tangles? Can we get tangles? And what we were able to see was, sure enough, if you wait till about 8 weeks, when you have the amyloid form in these dishes, then you start to see the tangles. See these little, in the middle, my pointer's dead, but in the middle panel, what you're seeing is nerve cells with that black inside. Those are tangles, and you see a magnification of that on the last panel on the right. And what's happening there is tangles are forming at about 8 weeks in this dish. So the next question was, are these really tangles? And if they're tangles, if you look at them with an electron microscope where you can see really close, they should look like what's called paired helical filaments, meaning they should look like these twisty things, almost like a helix. And when you do that, sure enough, this is what you see. This is from the dish. Normally, you'd have to get an Alzheimer's brain to see this. These are bona fide paired helical filaments saying that we've actually made tangles, not just plaques, in the dish. Then the question was, is the amyloid driving the tangles? And I won't show you all the data, but basically, if we used 4 different drugs that stop the amyloid from forming, guess what? No tangles. Stop the amyloid, you stop the tangles. So this was not only the first time amyloid and tangles could be recreated in a model of any type, mouse or otherwise, but in this case in a dish in just 8 weeks, we could also get the amyloid, stop the amyloid, and thus stop the tangles, providing the first proof of concept that if we stop amyloid early on, we'll stop the tangles, which means we stop the cell death, which means you stop the rest of the disease. And that's why this paper got so much publicity, in the New York Times and in the media, is because it's 2 things. One is the first full model of the disease with plaques and tangles, but second, after 30 years of conjecture, assumption, and debate, this says amyloid causes tangles, that the amyloid hypothesis is correct. It starts with amyloid. And the next question was, is it possible to stop the tangles even though you have the amyloid? And why I think that's important is that every once in a while, and David knows this, you get a brain that comes around from someone who didn't have Alzheimer's at autopsy, and you look in the brain and you say, wow, this brain's full of amyloid. How did they not have Alzheimer's disease? And the answer usually is they probably didn't have tangles. Is it possible to have amyloid without tangles? And in this case, we could actually use a drug that blocks a certain enzyme whose initials are GSK3 beta. When you block that enzyme, then you can have all the amyloid you want in the dish, you don't get the tangles. So for the first time in this system, we could basically disengage amyloid from tangles and show that for amyloid to cause tangles, you need that enzyme. That's just the beginning. Now we can test so many different genes, so many different compounds, so many different proteins, and ask, what is needed for amyloid to lead to tangles? How does amyloid lead to tangles? Here's a system where you can ask those questions, and experiments just take 6 to 8 weeks. In addition, you can start to do the drug discovery. So the answer to the question is yes, amyloid deposition leads to tangle inside of nerve cells. It requires this enzyme. And now what this means we can do is we can start screening drugs rapidly. So there are about, in the US pharmacopoeia, there are about 1,200 approved drugs that are used for all different purposes. And we've always wondered, I wonder how many of those individually or together might work in Alzheimer's disease. Wouldn't it be nice if we could test that? Well, in the past, we couldn't do it because if you wanted to test 1,200 different drugs against mouse models that take a year each, the amount of time and effort and expense was just prohibitive, especially doing combinations of all your 1,200 approved drugs on the market. Now with this, you can do them in a dish. You can do it each one 6 to 8 weeks in a dish. In each dish, we can do 96 drugs in each dish and just start running them through. And we can ask, what drugs already out there might stop the amyloid and thus the tangles? What drugs out there might stop the tangles but still let the amyloid form? This is exactly what Cure Alzheimer's Fund will now be funding, is this exact approach of taking drugs that are ready to go in clinical trials because they're approved and saying which ones might work here. We can ask that for the first time and do this at the right expense. We can also look at other drugs that are out there that are available, that already made it past phase 1 for safety, that thus have an easier route into clinical trials, and ask if they are able to stop the plaques and the tangles. So all this will be ongoing with Cure Alzheimer's Fund help. So I think if the original amyloid hypothesis was that amyloid leads to tangles, but they had all this junk in the middle that had to happen, well, no. We have to revise this. In our system, it says amyloid leads directly to tangles. You don't need a whole lot else to go on. And that means that we have to just ask, what are the actual molecular events in the cell that get you there? And then that will teach us how to prevent the tangles. So 2 things now. We want to not only prevent the amyloid early on, we can also start to think about how to stop those tangles from forming and spreading. I just want to mention in that regard that David Holzman, who's here, is doing some really cool experiments now with immunotherapy, but not using antibodies to amyloid. He's using antibodies to the tangles and trying to stop them from spreading in the brain. And we're really optimistic about his results that we'll hear about hopefully someday when he's ready. So that's a nice shot on goal for hitting the tangles. So this gets me to the final segment of how do we stop or delay all of these events, amyloid, tangles, and the inflammation? How do we get there? Well, we get there the same way we got here so far, which is you need evidence of who are the players. You can't understand how things work. I tell my students all the time, you can't understand how things work until you know what matters. If you want to know how something works, you have to find out who the players are and what matters. How has to follow what when you ask questions. And that means you're back to genetics. Genetics gives you the genes, it gives you the whats that interact to tell you how things work. So the Cure Alzheimer's Fund has supported, since its inception, the Alzheimer's Genome Project, which over the last 10 years has worked to identify all the genes involved that influence Alzheimer's. As it was mentioned by Jeff, the biggest whole genome sequencing project in Alzheimer's to date that we are just ready to complete and submit for publication, finding all the different mutations in these genes. And now the new phase, genes to therapy. Take all these genes, take these mutations that are involved that tell us what's going on, and turn that into therapies. How do we do that? And luckily, among the first genes that we and others have found, we can start to put them in these boxes. Genes that affect amyloid, genes that affect tangles, genes that affect inflammation. Each one of these genes is giving us a new insight into how these different aspects of the disease take place. We can look also at how these genes interact with each other. We can do what's called a systems approach and look at networks of genes that are all being activated together based on the Alzheimer's genes we just saw, that will give us an idea of which biochemical pathways, which biological systems in the body matter most, given the genes we found in these screens. And then once you do that, we can work with our colleagues like Eric Schott at Mount Sinai and say, given these genes, what does this tell us about which drugs already available on the market might be useful? And you start to get drug maps that look like this. And that will guide us when we do those 3D couches to say, these are the drugs we start with, because the genes in their network said these drugs are the ones that are most likely to work. So a lot of this work now becomes in silico, or systems analysis, once you have the genes that are at play. An example is getting back to the immune system. I mentioned about those microglial cells, that they can be very good or very bad. Well, this just shows you, so the green one on the right would be the good one. It's trying to clear the amyloid away. It's trying to clean up a region where there's been some cell death. The red one on the left is toxic. It's thinking it's protecting the brain, and the green one turned into the red one, and now it's shooting out free radicals and shooting out chemicals that create a very toxic neighborhood in the brain, all because it thinks that the brain's under attack. So we thought, wouldn't it be nice if we could know the genes and proteins that control whether the microglial cell stays a good cell or becomes a bad cell? And we found that out. One of them is CD33, which we discovered as an Alzheimer's gene in 2008 as part of one of the first results of the Cure Alzheimer's Fund Genome Project. The second one is called TREM2, which Dave Holtzman's been working on with his colleagues. And the easiest way to think of it is CD33 is the on switch that makes the microglial cells bad. TREM2 is the off switch. So now we can think about drug discovery of how to turn off CD33 or turn on TREM2 to keep those microglial cells chilled out and not attacking its own brain. And that's something we're actively doing. So this just shows you the two directions. So just to get to a closure here, and then we can take, hopefully, your questions. If we think about this original cascade of events where you start with the amyloid early on, then the tangles, the inflammation, the dementia, we can now think about what is the right stage to treat each patient. If you're going to use anti-amyloid therapies, you need to do that early on, and you can almost think about those like statins in heart disease, whether it's GSMs or immunotherapies or clearance drugs like PBT2. If you're going to try to clear tangles from the brain using immunotherapy, some of the work that Dave Holtzman's doing, you want to do that also pretty early on. But for a patient who has the disease right now, you want to do those things, but you also want to treat what's going on in that patient's brain right now. Inflammatory reactions, inflammation. So we need drugs that can protect cells from inflammation, so-called antioxidants. We also need drugs that can control those microglial cells and tell them to calm down, don't attack the brain, because that's what this pathology is telling them to do. And these are your three stages, and the idea is to have the right therapy, right patient, right time from prevention to treatment. This would be the goal going forward. And Cure Alzheimer's Fund is really leading the way here by showing us, supporting the genes you need to do this. And ultimately, someday, we want to have the mega 3D model of the brain. Most models are not cutting it. This is a new proposal from Hansong Cho, Du Kim, where we're going to go well beyond the 3D system we just published to have one that mimics the entire brain, from plaques to tangles, microglial cell inflammation, how the amyloid gets out of the brain through the blood-brain barrier, all the way to tangle spreading, and have this all in one dream model of a 3D brain model. This would be one of the goals of the genes to therapy. And we're already starting to do it in this gel matrix type system. So this is a little glimpse of the future of how we're going to have the ultimate 3D AD brain in a dish and be able to test all these different drugs at these different stages. That's our dream, that's our goal. And it's not that unreasonable. We're already on our way. And ultimately, we'd like to do this with a person's own stem cells. So if you're at risk, we take your stem cells, we turn them into nerve cells, and then grow them in this system and try to figure out what we need to do for you. That's our dream. It would sound like science fiction 5 years ago or 10 years ago. Now it's possible. All these systems can start to be developed to do this. This is our hope. Cure Alzheimer's Fund will be supporting it. So in closing, I think it's clear we're going to need a cocktail to treat this disease. We'll need a cocktail after this talk, or a glass of wine. According to the University of Bordeaux, red wine is very good for Alzheimer's disease. Here's proof. You get these two mice, and the scientist says red wine makes them live longer, but they get to be a real pain. And this little mouse is saying, it has a timid nose with the usual notes of oak and vanilla. And this mouse says, I can't believe you're drinking Merlot. So, in closing, I just want to, besides doing Alzheimer's research, some of you know I dabble with music on the side. This is playing with Joe Perry of Aerosmith on the Jay Leno show. You can see Joe up front here looking very savvy as a rock star, and me looking totally bewildered in the back on the keyboard. But I also like to write music. And recently, I teamed up with an incredible singer named Chris Mann. He just had a #1 adult contemporary album. He first got launched on the show The Voice, more of an operatic pop singer. There's a picture of him here on the right. And his dad has Alzheimer's disease, and we thought, hey, why don't we write a song together that basically captures Alzheimer's disease from the perspective of the patient. So this would be a song in which the patient is singing Remember Me to his loved ones. So I want to play that song for you as I end. And my hope is to use this song with the proper campaign so that all the profits from the song, if we get it to go viral with a video perhaps, will go to Cure Alzheimer's Fund to help support research. So you're among the first to hear this song. Chris Mann's on vocals. I wrote the music with him. We also have cello and violin on it, piano, and I'll play it for you now. Oops, that's not good. That wasn't good at all, was it? That was something. Okay, let me try this again, hold on. That's better.
I need someone to hold on for me, to what I can't seem to hold on to. The life we used to live is slipping through my fingertips like a thread that's unraveling. I suppose that nothing lasts forever, and everything is lost in its time. When I can't find the words that I'm trying to speak, when I don't know the face in the mirror I see, when I feel like I'm lost and alone in this world, won't you please remember me? Remember me. I know there'll come a day when I have gone away, and the memory of me will fade. But darling, think of me and who I used to be, and I'll be right there with you again. I hope I'm one thing worth not forgetting. Tell me that you'll never let me go. When I can't find the words that I'm trying to speak, when I don't know the face in the mirror I see, when I feel like I'm lost and alone in this world, won't you please remember me? Remember me. Remember me.
So on behalf of Cure Alzheimer's Fund, we will never give up. Thank you for your attention. Thank you. Thank you. Thank you. That's very kind of you. Thank you. So you can write your questions on the cards on the table and send them to the front. I'm happy to address them. I think we have some time to do this, right? So the first question is, with a family history of Alzheimer's, should I get genetic testing? So here's the thing. For Alzheimer's disease, if it's early onset Alzheimer's that strikes under 60, genetic testing is quite reliable. But for the cases, most cases, 95% of them that strike after 60 years old, there are, somewhere around 3 or 4 dozen genes, probably in the end 100 genes that can be involved, all interacting with each other. And the fact is we can't yet do reliable genetic testing. ApoE is the biggest of those, and we can tell you if you have an E4 variant that increases your risk, but that again doesn't guarantee onset of disease. You have to know all the other genes that work with it. So the answer is for early onset, yes, you can do genetic testing that's reliable. For late onset, we're kind of limited in our ability to do genetic testing and probably need to wait until we have better ways to do it. Another question is why have so many drug trials failed? And I think I hit that in the presentation, that we were targeting mainly amyloid. Amyloid's a very early event. We were treating patients way after the fact. So I think we just have to back up those treatments with amyloid to pre-symptomatic stage as is being tried now. Okay, now I have to try to read handwriting. Oh, okay. How limiting is the inability to control for the immune response in the study of drugs' effects on plaques and tangles? Very good question. Smart crowd. Must be at the Harvard Club. Yeah, so as we try drugs that hit early on plaques and tangles, it's a great question because everyone has a different set point at which time the immune system says, keep cleaning up, keep helping, get rid of the amyloid, get rid of that cell debris versus, oh my God, we're under attack, we have to fight. So this is important. That's why we're working so hard on finding now over a dozen different genes that control that set point of the immune system of the brain so we can get an idea of when a patient's immune system might kick in and start to confound the ability to stop plaques and tangles. But this is really a big concern for the future. It's a great question. Let's give me a chance to read. It's a long one. So what are the implications for the metal theory? So I talked a little bit about, not much, but I talked a little bit about how my lab back in 92 or so found that copper and zinc drive the aggregation of amyloid beta protein into amyloid aggregates and plaques. And we've been developing therapies, like I mentioned PBT2, that takes the metals away from the amyloid, in which case the amyloid can fall apart and be cleared away. I have a lot of hope for that because it's a very basic premise that metals do this, but I think like any anti-amyloid therapy, you have to do this early on. And if you don't do the right trial with the patients at the right stage of the disease, clearing amyloid may not be sufficient given that tangles are still spreading and inflammation is taking place. So if we think of a dream someday of knowing you're at risk, you know by imaging your amyloid's a little too high for your age, if you're 40 or 50, the idea of having a statin for Alzheimer's disease, whether it be a metal targeting drug like PBT2 or a gamma secretase modulator or an immunotherapy like solanezumab, this would be the goal, is to know your amyloid's a little too high, just like you might know your cholesterol's too high, and stop that disease at that stage before you get into trouble. So I think metals, targeting metals represents as good a strategy as any out there. What allows the tangles, et cetera, to grow so quickly in the 3D medium? Good question. These are great questions. Were these planted, Tim? Thank you. I love when he plants the questions. It makes it so much easier. In the case of the 3D culture system, you can ask also, why is it you can get amyloid plaques in the 3D system but not the 2D liquid traditional system? I think it's as simple as diffusion, right? So think about these nerve cells. They're making amyloid beta protein. If it's in liquid, it just disperses out, right? If it's in a gel, those little proteins move very slowly and stay around the nerve cell. They can find each other, form clumps and form plaques, right? And now these clumps stay in proximity to the nerve cell, and now they can attack the nerve cell, and that will cause the tangles to form. So I think it's a simple idea that Du Kim had to use gel, and it's probably nothing more than limiting the diffusion of the pathology so it can accumulate in just 6 weeks the way it takes 60 years or 70 years or 80 years to do so in your brain. And it's that simple. Let me see if I missed any over here. That one, I did that one. Okay. Would people who suffer concussions be more predisposed to contracting or accelerating Alzheimer's? Yeah, so concussions, any head injury, is an insult to the brain where you can get immediately tangle formation. So when we think about some of these unfortunate sports figures, football players who had more than their share of concussions, who 20 years down the line suffer from dementia, we call this chronic traumatic encephalopathy, CTE. And Lee Goldstein, my ex-postdoc who's now at BU, has led the charge on this with Ann McKee, and we had a paper together in 2012 showing that what's happening in an athlete with lots of concussions or in a soldier who suffered from a bomb blast, that you're instantly getting tangles. So the way to think about this is in Alzheimer's, the amyloid is slowly accumulating and causing tangles here and there, and it's like lighting a match and the tangles spread. In concussion, multiple concussion or a bomb blast, you're getting so many tangles in one spot all at once here, and then another hit and here, that they start spreading with multiple fires, so you get to dementia earlier. Inflammation's a part of that. So the tangles of the fire, amyloid is the most common match that starts it, that lights it in most people, and that takes decades. But if you're unfortunate enough to have enough head injury, you can get to tangles directly, and then those can spread over 10 or 20 years and get dementia. So I think that's the answer. I'll look at Dave Holtzman and ask him if he, as a reality check, yeah, okay. Got to always go to your teammates to make sure that you're not blabbering, right? So that's how I think it works. You're welcome. This person said thank you. Comment on the status of the timetable for FDA approval. Too long. Question is the FDA taking too long on approvals? I think the FDA has a job to do. Make sure that we're not hurt. Safety first, right? Health first. So we have to be very careful that drugs don't inadvertently have adverse effects and hurt us. So that's their number one priority. The drug has to be safe first, and then its efficacy has to be at least somewhat better than the original drugs that are out there. So I actually don't think they take too long. I think there's two problems in the system, okay? Everything's leading to personalized medicine, using genetics to predict you're at risk. If you know you're at risk, you do early detection of the disease, perhaps with imaging, and then you stop the disease before it strikes. So how do you do a trial for that? Just like the trial is going on, you have to have people who don't have symptoms of the disease, but who are already showing some of the early warning signs, and then you might have to do a 2, 3, even 5-year trial with a drug to say, if I give them this drug and they already have a lot of amyloid in their brain, but they're not showing cognitive symptoms yet, can I prevent those cognitive symptoms from occurring? That's a long trial, right? Well, drug companies are hesitant to do these trials because they're long and expensive. They were betting on whether amyloid would cause the disease. I think now, hopefully, with this new study, amyloid is shown to cause the tangles and the rest of the disease. There's still an issue, patent life, right? A lot of times, by the time you start a trial, there may be only 7 or 8 years, maybe 5 years left on a patent before a drug would go generic. So if you think about doing a 5 to 10-year prevention trial, or even a 3 to 5-year trial, by the time it's done and the drug's approved and on the market, it's already a generic. The drug company just spent billions of dollars and the patent's done. So I think that's an issue, is the patent law right now does not favor prevention trials. And of course, if you ask a congressman to help with this, they're going to have to tell the people in their state, we're going to make you wait longer for your generics. So it's a tough problem, and I think it's one we have to solve if we're going to do prevention trials. So I think that's an issue. The other issue with the FDA, I think, is they have to come around, and maybe our paper this week will help, to see that amyloid is to Alzheimer's as cholesterol is to heart disease, period. Okay, we already took the leap of faith and said, if you use drugs that stop cholesterol or lower cholesterol, you'll lower the incidence of heart disease, or at least diet or exercise, lower cholesterol, you lower incidence of heart disease, and more or less that's worked. We have to convince the FDA that now, and hopefully it'll be easier after this study, I don't know, that if we can stop amyloid, especially in someone whose imaging says they already have too much amyloid for their age, that they're going to stop Alzheimer's down the line. If they make us wait for 5 or 10-year prevention trials that drug companies may or may not want to do because they're too expensive or patent life runs out, we may never get there. So good question, and these are major issues going forward to make sure that we stop this disease. More questions? There are 3 secretases. Why only gamma secretase? Can we modulate the others? Somebody really knows their stuff. Who wrote this? Is that you, Dave? Okay, somebody's savvy. Yes. APP, so if you picture this big protein this long, and in the middle of it there's this little segment, this the amyloid beta protein, there are 3 different cuts, right? Normally, something called alpha secretase cuts in the middle of the amyloid beta protein, so you can't make amyloid. That's the most common cut. We just showed the gene for that, ADAM10, has mutations that lead to late onset Alzheimer's. To get amyloid, first you have to have beta secretase cut and then gamma secretase cut to excise it. And in fact, there are trials for both. Beta secretase inhibitors are being tried by several pharma companies, I think 11 of them, and gamma secretase modulators, which Cure Alzheimer's Fund is supporting, are now just going into clinical trials for the ability to just specifically stop gamma secretase on amyloid. So we're actually hitting beta and gamma. You can ask, why aren't you trying to enhance the alpha secretase cut, the good one, that precludes the amyloid? Well, it turns out that if you have too much alpha secretase activity in your brain, it's not good. In mouse studies, it's really toxic to have too much of that cut. So even though it'd be nice to have more of it and have less amyloid, the other activities of having too much of that good cut with alpha secretase just would be toxic, is what we think. Okay, heading the ball in soccer sounds disastrous. Why are we encouraging soccer? So I played soccer in high school and then for a while until college, and then I started working in a lab, and that was it for soccer. You know, if you have a brand new soccer ball that's fully inflated and it's a sunny day, unless the kick is very hard, it's not too bad, right? What I worry about is what happens most of the time. You're on the practice field, you're using a practice soccer ball where all the polyurethane is gone, so it's like a big sponge, and it's raining out, and this soccer ball that's 10 years old is soaking up water because there's no polyurethane cover on it, and it weighs about 5 pounds. And now somebody kicks it 50 yards in the air, and you say, I know, I'm just going to hit it with my frontal cortex. That is a bad idea. So I share this concern. I don't want to see sports stop. Soccer's a great sport. Football, I mean, it's such a touchy thing to talk about, but it is a concern. Helmet-to-helmet crashes, head bangs, hockey, football, heading the ball in soccer. I have a little girl who doesn't like sports. I'm very happy about that. But yeah, this is a concern going forward. It's going to see how it plays out, but it is a real concern. Funding. How do you see funding and a timetable for gene set therapy? Glad you asked. Was that planted? Yeah, so gene set therapy, this is a major campaign right now. We want to do everything from what we can do in mouse models to understand how the genes work by putting these new Alzheimer's genes into the Alzheimer's mice and seeing how it affects amyloid deposition and other aspects. A big focus will be on these new 3D systems, getting to the ultimate 3D Alzheimer's brain, and turning that into drug discovery. It's going to be expensive. It is going to be expensive. It's going to be millions of dollars. So we're fundraising for it. We're already going. Cure Alzheimer's Fund very, very wisely had money in the bank ready for this. It's ready to go, and we're ready to use it, and we're using it. But it's going to take a lot of work, a lot of time, a lot of money, but I think it's going to be well worth it. Last one. It says last one on it. Thank you. You're welcome. Thank you for the song. Yes, I didn't mean to bum everybody out with that song, by the way. I hope I didn't. This is mainly a thank you to Cure Alzheimer's Fund, and saying that we need to work harder. And all of you here is really evidence that we all care together, and we're going to do whatever we can together to stop this disease. And the Cure Alzheimer's Fund, I can't say enough good things about them. They're the ones who really deserve the applause here, our founders, our board, staff members. So thank you very much once again. You're too kind. Thank you very much. Thank you. Thank you.